Hi everyone, this is lesson 24. Today we're going to be talking about the ETC, or the electron transport chain. Our lecture outline is as follows. We're first going to look again at metabolism as a whole and see where the ETC fits in. We're going to have several lecture objectives, list and describe the components and roles of the complexes in the ETC, describe the structure and protein organization in the IM of the mitochondria. We're going to look at the free energy of electron transfer and identify the source of the protein gradient. So let's begin. First, let's start with a picture of the mitochondria. This you will have seen already. Recall that we have already talked about glycolysis, which takes glucose and converts this to pyruvate. This is in the cytosol. Recall that we have talked about the conversion of pyruvate to acetyl-CoA. This is the PDC. And also recall that we have talked about the citric acid cycle, or the TCA. So now we are looking at the conversion of these reduced coenzymes, such as NADH and FADH2, and how we're going to get energy in the form of ATP from these reduced coenzymes is through the electron transport chain, as well as ATP synthase complex, which is going to be our next lecture. So, important take-home points here are we're in the mitochondria, we're at the end of our kind of metabolic stretch here, where we've started from glycolysis, then to PDC, then to citrate cycle, or TCA, and now this is at the very end where we're going to convert these coenzymes to ATP. So electron transport in the cellular context is the oxidation of these reduced coenzymes, NADH and FADH2, and this is carried out by the electron transport chain, or called ETC. So for two important points here to start off with. The first is that electrons are passed through a series of electron-carrying molecules, and the passing of these electrons is based off of this standard reduction potential. So we will see that in the coming slides. Number two, electron-carrying complexes, or these electron-carrying molecules, are ordered in the inner membrane from low to high standard reduction potential. And we're going to see why this is a very, very important property in the next slides. So a little bit of intro and background to start off with. The figure on the left is a little bit small, but it's basically a picture of the mitochondria shown here, an experimental snapshot of the mitochondria. And then shown below here is inside the inner membrane wall are the different complexes. These are the energy carrying complexes that we're going to talk about. So let's have a quick review as to where we're at after glycolysis and TCA in terms of the number of reduced coenzymes and high energy compounds. From glycolysis, we have 2 ATP and 2 NADH. From the TCA, if we include the PDC reactions, we are at 8 NADH and 2 FADH2. We also have from TCA the GTP, which is similar to the ATP. So we do have some high energy compounds as well. So our total at this point, if we sum up all of this, is 10 NADH, 4 ATP, and 2 FADH2. So this is a very important sum total for you to know and be familiar with. So if we take all of these coenzymes and high energy compounds and we ask the question, how much energy or energy return does the reduction of these coenzymes yield? We would see that this yields about 40%. And if we compare this to alcoholic fermentation and homolactic fermentation, recall that This is coming back from around the time where we were learning about glycolysis. This only yields about 9% to 11%, so obviously much more efficient. So now we're going to see how all of this works through the electron transport chain. ATP production we will talk about in the next lecture, and remember that all of this is still occurring in the mitochondria. So where we are in the cell in terms of the ETC is the mitochondria. You should be familiar with the different parts of the mitochondria shown here. So we have an outer membrane, we have an inner membrane, and then between those two is an intermembrane space. We have something called the crista, which is shown here. The mitochondrial matrix are all these folds that are within this inner membrane. And finally, shown here, all these little spots are the ATP synthase complexes that are going to be producing ATP that we're going to talk about in the next lecture. So one of the important properties of the mitochondria is the mitochondrial matrix, and the fact that there are these folds means that we're going to have a very significant increase in surface area for the components of the ETC. So we'll be able to have many, many, many more protein complexes be able to fit inside the mitochondria thanks to the folds of this molecule. So the more of those complexes we can fit, the more ATP synthase molecules we can fit, the more ATP the cell can produce. So I was saying that the ETC components, so these different complexes that we're going to be talking about, they are ordered in the inner membrane of the mitochondria from low to high, standard reduction potential. So that means over here is where we're going to have a high standard reduction potential, and over here at the beginning is where we're going to have a low standard reduction potential. And so basically you can see all of the reactions. These are all going to pass these electrons from complex to complex, starting from complex 1 to complex 2 to complex 3 to complex 4, and then we have our ATP synthase complex, which is going to then make our ATP, as shown here. We then have a ATP versus ADP translocase that pumps that ATP out of the membrane so that it can reach other parts of the cell. So if we look at this, all of these different ETC components, let's just go through in kind of coarse detail what's really happening here. So as you can see, complex 1 is going to be the start of this. This is going to be the lowest standard reduction potential of all these complexes in this series. And here we have an NADH being oxidized to an NAD+. So here we're going to have these 2 electrons that are taken from the reduced form of this coenzyme, and these are going to pass through complex 1. And see how our 2 electrons are then going to pass through complex 2. And we have another reduced enzyme, FADH2, being oxidized here. So then we have what's called complex 3 that's going to continue to pass these electrons, and a cytochrome C molecule that will also be helping out. And these electrons are going to keep passing through complex 4 now to where we finally have a oxygen molecule that's waiting, and we're going to make water. And we're going to see that in the end, all of this is to establish a proton gradient between the mitochondrial matrix and the inner membrane space here. So note here that we have 2 protons that are going to be pumped out. We have 4 protons that are going to be pumped out, and another 4 protons here that are being pumped out. So we will see why this is all important, but these are the important aspects to keep in mind right here. So just a quick summary of this. Our complexes are organized from low to high standard reduction. They're organized also by number. So 1 is going to be the lowest, 2 is the second lowest, 3, 4. These are all going to be in order in terms of low to high standard reduction potential. And then we're going to see our reduced coenzymes that get oxidized, and these electrons are going to pass through each of these complexes from 1 to 2 to 3 to 4. In the end, an oxygen molecule is going to take those electrons, and then in the next lecture, we're going to see the end product here of how ATP synthase is going to work. So the standard reduction potentials, this is a table that shows us the electron carriers in this electron transport system. And if you look at this carefully, and you can see as we go from the NAD plus to NADH, all the way to our oxygen that's going to then be converted into H2O, there's going to be a low to high trend here. So take a look at complex 1. We have negative 0.25. The coenzyme Q is a part of this electron transport chain. But before that, sorry, I missed complex 2 here, which is also succinate dehydrogenase from the TCA. So here we have our FAD that's going to be enzyme bound. And the whole kind of total standard reduction potential for this complex 2 is now negative 0.04, so less negative than the previous complex. Now, if we look at coenzyme Q, which is also a part of this series, we're now at around plus 0.04, and our complex 3 is right around the same. Now we're at the cytochrome C, which is another part of this ETC, and we're now at 0.21, plus 0.21. And now all the way down here, where our oxygen molecule is going to take those electrons, this is now plus 0.82. So notice here we have this negative 0.25 all the way to positive 0.82. So we have this trend of low standard reduction potential to high standard reduction potential. So this is another way to look at the same thing. So these are the standard reduction potentials of the major respiratory electron carriers. Let's go over these axes first. So we have our energy, our standard energy in terms of volts. We have our delta G standard in terms of kilojoules per mole, and this is relative to oxygen. And what is plotted here across these two axes here is as we move from complex 1 all the way through the electron chain to oxygen. So what is shown here is the different steps of this trajectory or this reaction coordinate of all the different complexes in the ETC and how energy is going to be released in terms of how exergonic it is as these electrons move from the reduced coenzymes through the different complexes of the ETC to the final molecule of oxygen. So shown here is NADH, and we have this moving to an FMN, which then goes to a CoQ. So this is all these are electron carriers. And as NADH passes its electrons to FMN and CoQ, then we're seeing that this has a negative 69.5 kilojoules per mole associated with that transfer. So now that we're at CoQ and we have a transfer to cytochrome B to cytochrome C 1 to C and to the A, we see another decrease in the delta G of negative 36.7 kilojoules per mole. So again, just keep in mind, these are all just different electron carrying compounds that are embedded in the ETC. So as we move now from cytochrome A to cytochrome A3 to finally our oxygen, which is the final electron carrier, we get a pretty large decrease in our delta G, which is negative 112 kilojoules per mole. So because of the properties of each of these electron carriers, they're going to have different standard reduction potentials. And as these are organized from low to high, the cell takes the property, this particular property, that takes advantage of it in order to have the process, the whole process of transferring electrons from NADH to oxygen as an exergonic spontaneous process. Not only is it an exergonic spontaneous process, but it's very controlled. So you're not going to go directly from NADH all the way to oxygen, and we'll see why that is in the next coming slides, or especially in the next lecture. We want to take advantage of these individual small increments in order to basically create a proton gradient between the inner membrane and the intermembrane space. So these are the important properties here to remember, that they're organized from low to high standard reduction potential. There are a number of these different electron carrying compounds that each have a unique standard reduction potential, and the way that they are placed physically in this membrane has a functional role in transferring these electrons from low to high standard reduction potential eventually to oxygen, as well as having incremental steps to secure this proton gradient. So we're going to watch a video. The video is really helpful to have animation to see how this whole process is coming together. So from this video, there's 6 take-home messages that I would like you to keep notes on, and I've outlined them here. So the first is to look and think about when this proton gradient in the matrix is talked about, what is talked about for this proton gradient. The second is looking at the ATP synthase, which we're going to talk more in detail about next lecture, the importance of the rotation for this complex. Number 3 is looking at these 4 proteins and complexes of the ETC, 4, understanding what redox centers are. 5, looking at complex 2 in particular and how this is unique. This complex 2 has a FADH2, and there is also coenzyme Q. And finally, the importance of oxygen. So let's see this video. I may have to come out from this screen for a second. Okay, so I'm going to mute this and I will narrate on top of this for us. So let me, okay. So this is a video that has been put together by Harvard. So these are eukaryotic cells from yeast, and they contain the membrane-bound organelles with specialized functions. The mitochondria are double-membraned organelles, and these harness the most energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial matrix. So this is an image of the inner mitochondrial membrane, so all of your lipid bilayer there. There you have the lipid tails versus the lipid heads. And one of the key roles of this membrane is to act as a barrier to positively charged protons. These are allowing a concentration gradient to be maintained where the inner membrane space has far more protons than the matrix does. So look at how much the volume of those protons are versus inside the matrix. The membrane also contains a large proton complex called F1, F0 ATP synthase, and this uses that proton gradient to drive the synthesis of ATP molecules. ATP molecules are then providing the energy for most of the cell's reactions. Just as a man-made power plant produces electrical energy using the flow of the wind, water, or stream to rotate a turbine, this synthase molecule makes ATP by using that proton flow from one side of the inner membrane to the other in order to rotate those protein subunits. If there's no gradient, the synthase subunits will stop rotating, and then because the cell becomes starved of energy, it will die. So the protein complexes and the small molecules that establish the gradient and maintain it are playing an essential role in the life of the cell. At the heart of this system are these 4 protein complexes in the ETC, numbered 1 through 4. This is what we've been discussing here. And these directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complex 3 and 4. And proton pumping requires energy, and the 4 protein complexes get this energy by transferring these electrons through a series of reactions. So this is the linked process of electron transport, which is why the 4 complexes are referred to as this electron transport chain. So their processes are directly linked. So we're going to focus on complex 1. This is a byproduct of sugar metabolism. So NADH deposits 2 high-energy electrons in complex 1, and they're passed along a chain of redox centers. Remember the redox centers we talked about, where they were structured from low to high standard potential. So these redox centers have different affinities for the electrons based off of their unique atomic coordinations. So let's look at a couple of these and reveal 2 reasons why the electron moves from the top to the bottom. First, the bottom redox center has a higher affinity than the top one does. This has to do with the standard reduction potential. Second, the distance between these is ideal for the electron to jump, and this explains why electrons don't bypass the bottom redox. They go through each and every step of this chain. A small amount of energy is released each time an electron is passed between the redox centers. And complex 1 harnesses this energy across all the redox centers and uses it to pump the protons across the membrane. So the last redox center in complex 1 will donate the 2 electrons to coenzyme Q. And that will take those 2 electrons through complex 2 to complex 3. Complex 2 is similar to complex 1 in 2 important ways. There are high-energy electrons that also enter complex 2 via this byproduct of sugar metabolism, although here the molecule is FADH2. And the complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. A major difference, though, for coenzyme for this number 2 is that it does not use the energy liberated to pump the protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. And one electron is recycled and can re-enter complex 3 later, but the others pass through 2 redox centers before reaching cytochrome C. Cytochrome C then carries the electrons to complex 4. The electron transport chain then ends in complex 4, where a series of reactions involving 4 electrons convert a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored that each protein complex in isolation, but in reality, they are very, very densely packed. Together, they effectively make up the inner mitochondrial membrane, a giant cellular power plant. Okay, so now I would like you to think about the questions that we've outlined here, and we're going to go over those together. So we have a proton gradient matrix. This functions to produce a potential, where we have protons as the most important part of this. This is establishing the rotation of an ATP synthase, which is incredibly important for making those ATP molecules. We have 4 protein complexes of the ETC. They are explained individually, but they work together as a system and couple the pumping of these protons to making ATP and also carrying these electrons. We have different redox centers that have unique atomic configurations with different affinities for attracting and donating electrons to other centers, and the complexes make use of the small energy that's released from going 1 to 1 to 1 redox center in order to pump the protons into the matrix, the membrane, across the membrane, We also have a complex 2 that is slightly different in that it's taking in FADH2, transferring those electrons to coenzyme Q. There is no pumping, but these electrons are donated to the complex 3. And finally, our oxygen 2 is our final electron acceptor at the end of the electron transport chain. With that, that is the end of this lecture.